Over to you now. Did you hear me, Neza?
Okay, try not to. Hi, can you hear me?
Yes, I can hear you.
Okay.
So I was saying,
sorry, Lil, I tried to switch to the Bluetooth AirPods, but it doesn't work.
Oh, okay, no problem. I was saying I'm handing Prof over to you. You will handle everything, introduction, and then the start. I think we already have 26 participants, which is really good. We can start.
Okay.
Okay, great.
So, hi everybody, and thank you for joining us for this webinar today. I had the privilege to present Professor Bobit Badawi, who is a colleague of mine. So, Professor, sorry.
You are freezing now.
Neza, I think that, I don't know if it's you or Prof who has a feedback.
You have what?
There's like a feedback in the background. Maybe
I don't know, really.
What do you mean? A sound background?
Yeah, a background noise. But it's fine. You are audible. You can continue.
Okay, I don't know what I can do.
No. Just go ahead. We can hear you.
Okay, perfect. Thank you. So, as I said, Professor Badawi is an expert in genetic.
Okay, guys. I think Neza seems to be freezing a bit.
Yes, I think she has problem with connection. Yeah.
Yes. So I think what we're gonna do, so that we don't waste time, Professor Badawi, can you just quickly introduce yourself and then we can move forward? Neza, you seem to be having a connection issue. So I'm just going to allow Prof to go ahead and introduce himself, and then we can start. Anything missing, if your connection is better, then we can move forward at a later stage, just to save time. Thank you, Ned. Thank you, Prof. Please go ahead and show us your screen and give the presentation. Thank you so much for joining us. We are so excited to hear from you.
Thank you very much. Thank you very much, Lile. So my name is Barbit Badawi. I'm professor of genomics and bioinformatics at Mohammed V University in Rabat. I'm mainly working on biodiversity, but using either structural genomics or transcription genomics or proteomics. I'm North African coordinator too of Africa BP project, which is, I guess, the biggest project of sequencing in the continent, and which I will take the advantage of this presentation to share with you some information about this project. And I'm working too on making animal breeding schemes, combining genomics and artificial intelligence. So it's mainly, this is my domain of expertise. And I'm really happy to be invited in Sonar webinars. Thank you. Lile, could you hear me?
Yes, Prof, we could hear you.
Prof, we can hear you. You can go ahead.
So should I go ahead with the presentation?
Yes, please go ahead. And we can also see your screen. All you need to just do is put it on presentation mode and we'll be good.
So tell me, please, if you can see my screen.
Yes, I can see your screen.
Okay, so hello everybody. So hi everyone. So again, my name is Barbit Badawi. So I will try to speak about genomics studies and into application neuroscience. But before, as I mentioned in the introduction, I will start by describing the African BP project, which is project of sequencing of all the eukaryotic species in Africa. And in the second part, I will speak about genomics in neuroscience. So I think it's very important to speak about the African BP project because first, it's the biggest project of sequencing in the continent. And second, which is the most important, we would like to construct synergy between the African communities working on different aspects of biology. So let's start by speaking about the Africa BP project. So Africa is very rich in terms of biodiversity. It's home to 52,000 to 72,000 plant species, a quarter of the world mammalian and bird species, at least 2,000 fish species, and 100,000 species of insects, spiders, and other arachnids. And thousands of those species are endemic to Africa. Global biodiversity loss is happening at unprecedented scale in all over the world, Africa included. But specifically, this situation um, could be very dangerous for Africa, as 95% of the continent's known endangered species are not yet sequenced. And when we don't have this information of sequencing about the endangered species, it will be very, very hard to conserve the species or to find any solution that could save those species. So in the past, two revolutionary global projects have been done. Some are ongoing. The first one is the human genome that revolutionized the understanding of the human health. And second one is the biogenome that will improve our understanding of the biosphere. Unfortunately, Africa left out of the global genomic efforts, either for the first or second of the other projects. I will speak about some reasons behind this. So, Africa is lagging, as I mentioned, behind the rest of the world in developing and using genomics in medicine as well as in animals and plants because few or non-genomic efforts has been deployed to sequence a human or non-human species. Around 70% of the projects that targeted biodiversity have been led outside Africa, more likely with scientific agenda different from that of the African population. Because, of course, any funding comes with an agenda, and the agenda looks at the people that fund the project, not the people in which the projects are done. So furthermore, almost all of the sequenced species in those projects have been sequenced outside Africa, missing the opportunity to establish genomic platforms within Africa. So as mentioned before, as the scientific project really start from within Africa, the real science agendas of the continents are often, of the African continent, are often neglected. Here is a concrete example. Uh, in the context of the Human Genome Project, less than 2% of the analyzed genomes during the 2 decades since the project began are from African individuals. Even though, as we know, Africa harbors more human genetic diversity than any other continent, as it is the cradle of the humanity. The African researchers who contribute to the data collection in such projects are not always credited for their work, as providing samples is often considered a marginal task. So in this context, we have seen the birth of new promising initiatives, which is the Africa Biogenome Project, which intend to, first, coordinate the sequencing of all non-human genomes in Africa. Second, initiate and develop support to the sequencing of more than 1,500 species in Africa. And third, deploy the genomics information to inform policy and build capacity in Africa. So it's the first project in Africa that is done by African and for the African continent. So those 1,500 species that I speak about will cover all eukaryotic lineages, as we can have a global genomic representativity of all eukaryotes in Africa. So to ensure satisfactory inclusion strategy, Africa BP, the project Africa BP, is employing strategic partnership framework, including regional nodes, national ABC, public and private labs within the African countries, even even the foreign labs within African countries, and the big sequencing companies in the country. And the country is divided quite to four regions: North African, the South African, East African, the West African, and each region has its node and its IBC and its representativities. Actually, Africa has more than 200 genomic capacities. Unfortunately, 71% are concentrated in 5 countries, including South Africa, Kenya, Nigeria, and Morocco. And there are very, very small other capacities distributed through the African continent. Africa's internet, among the challenges that we have in Africa, is the cost of internet. Because Africa's internet costs are the most expensive globally. Look, for example, this is just an example. If we take Zimbabwe, the price is $75.2. How it is, it's $1 in Finland or $12.4 in the United States. So this is the cost for 1 gigabit of mobile data. So this is a big challenge concerning the internet costs, which may hinder establishing computing capacities within some African countries. So yes, there are many challenges to Africa BP is facing, but as you know, there is technology, it is here to help. For example, hybrid mobile reference labs is of great relevance to Africa and could be part of the solution that Africa BP could adopt. So to conclude about these parts, so Africa is left out of genomic efforts undertaken in the last 2 decades. However, the game is not over yet, as initiatives as Africa BP could generate capacity building of African scientists, genomicists, and bioinformaticians, could have scientific, technological, and socioeconomic impact, could impact the environment and African landscape, and could increase scientific cooperation between African countries. So the scope of this presentation of the African Biogenome Project is really to make all the African scientists aware of such initiatives, because we believe that the data generated by genomics and generated by the sequencing could be of great use to all the scientists for the African countries. And some collaboration could be done, some interaction could be done between the different scientific communities to orient the sequencing and to target the sequencing, which is already started by specialists in agricultural science and agronomic science and biodiversity science, but it could be done too by people working in humans, either in neuroscience or other disciplines of humans. So I will start now to speak about our main project, which is genomics for neuroscience. So Let's start by asking the question, what are genomic studies? So genomic studies are the study and analysis of an organism genome. It concerns the study of all the genes, either using DNA, RNA, proteins, or metabolites. Metabolites could include sugars, nucleotides, or others like lipids. So targeting one of these molecules, it depends on the scientific question we are asking. So when we have question about structure of the DNA, we use DNA. What we need, transcriptomics, or the gene expression, we use RNA, or the protein expression, we use the proteins. Some cases we use one approach, in others two, and in others we combine all the approaches. It depends always on the scientific question we are putting. So what is the difference between genetics and genomics? Of course, it's a difference of scale. Genetics is the study of heredity, which concerns one or few genes. So, and we are mainly interested in understanding heredity or understanding the function and composition of one single or few genes. While genomics, it's interested in all the genes of the genomes, and it addresses all these genes in the interactions, either using, as I mentioned before, the DNA, the RNA, or the proteins. That the most important is to tackle all the genes at the same time. So when we are using genomics, there is a shift of paradigm, which make many biologists, including neurobiologists, reluctant and skeptical to adopt this approach. One of the reasons behind this reluctance is what the aversion to what we often stated fishing expeditions, because those scientists think that when using omics, genomicists generally have no idea about the hypothesis they are asking. Yet omics, or what we call discovery-based approach, do not avoid or eschew hypothesis, but rather they seek to elevate hypothesis testing and to allow high throughput hypothesis generation and prioritization. What does it mean? It does mean before doing omics, I may start with one hypothesis, but when I get massive data concerning all the sequence of the genome, or all the transcriptome, or all the proteome, or all the combination of all, I might change my hypothesis, or fine-tune my hypothesis, or moderate my hypothesis depending on the data. So for that, we name that discovery hypothesis. So this is paradigm shift. Sometimes it's problem for traditional biologists for using this new approach to answer the scientific question. Apart from this paradigm shift notion I explained before, there is other challenges or reasonable challenges in neuroscience, and to explain why neurobiologists specifically have been slow to embracing genomics, at least comparatively to cancer geneticists or other biologists. These reasons include extreme cellular heterogeneity, complexity of known circuit, and reliance of post-mortem material. Another obstacle, which is consistent obstacle too, is the generation of enormous data amount. As the generation suppose the integration of computational biology or bioinformatics in neuroscience laboratories, or grouping with scientists with different specialties, including biostatistics, bioinformatics, genomics, and neuroscience in this case. Of course, this is challenge to the traditional way of doing biology generally, or neuroscience globally. But this will become more critical as more powerful technologies that are like next generation sequencing, that are generating many orders of magnitude more data, is now function is in place. So it's more challenging not to use all those disciplines. So let's jump to some application of genomics in neuroscience. Of course, genomics in neuroscience, or what we know neurogenomics, so could use many approaches of genomics, as I mentioned before. It could be structural genomics by searching for SNPs or the structure of the DNA, transcriptomics by measuring the expression of RNA, proteomics by measuring the expression of proteins, epigenomics by looking to the methylation or acylation of the genome. So there are many applications of genomics in neuroscience. Those applications have been done in the last years and has been highlighted by several successful studies to identify, for example, the molecular basis of neuronal diversity, the synaptogenesis, the finding biomarkers for some diseases, and finding mechanism and pathway for the diseases, and the development of genome scale resources. So, and more now, we are clearly there is we are weakening or we are making weaker the notion of the superiority of research based on single hypothesis testing over hypothesis generation and prioritization using omics based or discovery based methods. So we are more and more convinced, or at least the neuroscientists that are using genomics, that are more and more convinced that using this omics data and this massive data from which we can do a comeback and change our hypothesis or adjust our hypothesis is a stronger approach to use, and it's more likely better than the traditional way of doing hypothesis, starting with specific hypothesis and do some traditional experiment to get the answer. And this is if we look to what has been done in some disciplines of biology that use a lot of omics data, like cancer biology, we can see that when we started to use this high throughput approaches, we start to get some practical results, like by disease markers, like therapogenics, like many other practical advancement in this field of biology. So, of course, genomics is one of the strongest point of genomics is that it gives us an open database, and this open database include genetic information or genomic information. It could be DNA, RNA, proteins, or any other genomic data. And those data are often or goes often beyond the scope of their original experiment. So they are available to the scientific community where they can be mined or combined with other studies. So here I just give some example of those databases, which include NBL, ADB, NCBI, and many other databases. So those are general databases, but there are some specific databases, some for neuroscience. So let's speak about some specific databases for neuroscience. The most important one is those that try to survey the nervous system transcriptome. So the knowledge of the spatial, of the difference of spatial and local and temporal expression of every gene in the nervous system, it's a natural extension of the Human Genome Project. So a number of studies have been undertaken to identify neuroanatomical patterns of expression in the nervous system. And I think among the most important databases, there is the Anil Brain Atlas. Here we have the website www.brainmap.org. So which is a neuroanatomical repository of gene expression information in both mouse and human brain. So, and they contain the information concerning the gene expression according to the time and according to the space. So we can use this information to we can use this database to get information about which gene in expression in which part of the nervous system. So the users can query the maps by gene or brain structure and generate three-dimensional overlays of any permutation of gene and structure. And there are, this is one, but there are many, many other databases here. I cited GenPaint, Vision, GenSat. There are many, many, many databases that contain gene expression data for neuroscience. And it could be very, very useful for the population that are working or that would like to understand some aspects of the neuroscience using genomics. Another application of genomics in neuroscience is the exploring of the synaptic proteome. So you, as you know, and you know better than me, synaptic transmission is fundamental component of the nervous system function, and its dysfunction is implicated or responsible from for many, many neurological and psychiatric disease. So, thus, identification and function annotation of the molecular components of the synaptic proteome is very, very important. We can, so at least we scientists have to explore three compartments: the presynapse, the postsynapse, and the synapse, the matrix synapse between the two. So to get information about this protein, of course, we cannot do that using gene expression. It's better to do it using proteins because gene expression is not that good to provide a clear picture of organ-specific data. So we need more likely proteins than using gene expression. Of course, it's not that an easy approach because rich in genomics to understand the expression of proteins within synaptic, we need uh, a long-term approach which typically involve laborious purification of different synaptic components using immunopurification, affinity purification, subcellular fractionation, taking advantage of the different density of different organelles, after which we can use mass spectrometry and we can get the data, the protein data, or the expression of the protein in each synaptic compartment. Another application of genomics in neuroscience is the integration of genetic and phenotypic data. So, normally, what we do when using genomics to answer some questions, we can use the gene expression and we can use the SNPs or the polymorphism in the genome, and we can link the SNPs to the disease or we can link the gene expression to the disease. But we can associate these two approaches. I mean, associate the gene expression with the polymorphisms and the mutation in the genome to get insight of some diseases. Here is an example of the Alzheimer's disease. I have two groups here, the controls, the Alzheimer's disease and the early onset of Alzheimer's disease. And here we have the gene expression within each group of the three groups mentioned here. And in the same times, we look to the SNPs of the people that were introduced, that were used in these studies. And we found that there are two SNPs, SNP1 and SNP2, that are different between those populations. So what we can do is to associate the gene expression between the SNPs and to see if there are some SNPs that are responsible for the difference of expression of genes between these three groups. So if we associate these two ones, so gene expression and SNPs, just concentrating on this yellow rectangle that represents the genes that are differentially expressed between the two groups, we can see that the gene expression is in the y-axis and the genotype in the x-axis are correlated. I mean, there are some SNP, specifically SNP1 and 2, are responsible for the high expression of genes in some individual and the low expression of genes in other individuals. So in the future, instead of repeating the experiment and measuring all the expression of genes in all the individuals, we can just use this SNP as a proxy to deduce the gene expressions and to have an idea about the phenotype in this case, the Alzheimer disease in those individuals. Another application of genomics in neuroscience is a molecular network. We have many applications, just here I cited some. For example, we can take the gene expression and we can construct according to the expression of genes in those different groups. We can have a clustering of different groups. Here, for example, group E, B, C, and within each group, we have some genes that have the same pattern of expression. So we can detect which genes have the same pattern of expression. Another applications, we can have a hub of genes by using networks. We can have hub of genes which are the most connected or central genes. And from this hub of genes, we can depict the most important genes. For example, here H1, H2, and H3. Why are much important? Because those genes are responsible for the expression of other genes. So if in the future I would like to make study about gene expression in some diseases or some biological phenomena, I may be studying this genes here, or this model genes here, will make things easy for me, because I know that the expression of these genes is correlated with expression of many other genes. Another application here is the model relationship. In this, the multidimensional scaling plot here, principal component 1 and principal component 2, in this plot, we plotted the gene expressions of different cell lines and of different tissues or in the nervous system. And as you can see here, just by using the gene expression, we can clusterize all this tissue of the nervous system using just the gene expression. So when we have the gene expressions, we can have a clear idea about the phenotype, either here about the tissue or about the most important genes involved in the expression of the other genes, or the cluster of genes that have the same pattern of expression. Another application, which is now, the new trend of above all by the development of artificial intelligence and machine learning, this is data integration. I mean, the traditional experimental approach to the complexity of the neural system normally uses one layer of information, one or two layers of information. So typically we use DNA data, what I mean by DNA data, polymorphism, SNPs, or we use gene expression data, I mean messenger RNA, gene expression data, to understand problematic related to the neural system. But this approach seeks to consider all of these aspects at the same time. For example, here, I mentioned phenotypic data, gene expression data, epigenetic data, copy number data, sequence data. By putting together all this information and combining them in a mathematical way, which is now easier by the development of artificial intelligence, this facilitates the connection between different types of information. And here we have an illustration of the potential system-level integration of original brain gene expression, which was coupled with network-based analysis using imaging data. This was in our scope to provide insight into brain connectivity. So the identification of this brain connectivity illustrates the potential system-level integration of regional brain expression. And this is a successful example of how we can discover how different regions of the brain are connected. This is the lines in red and green highlight the connection between different regions of connections. And this is, if I remember, was done for data for language. I mean, the combination of diffusion tensor imaging data for the language with gene expressions, and they used this approach of network analysis using data integration, and they were able to highlight how the different regions of the brain they connect with each other. So here I just give some examples of how genomics could be used to understand complex questions in neuroscience. So I hope it is useful. Thank you for listening, and if you have any questions, I will be happy to try to answer them.
Thank you so much, Professor Gurjit Badawi. Thank you for this very interesting presentation and webinar. And now we open the session for your questions.
Sorry, Nasa, can you please repeat? You're not so clear.
I just thank Professor Badawi for the presentation and requested for any questions from the assistant.
Just in case there are other people who may not have heard Professor Neza, she was just thanking Professor Kouabit for an amazing, excellent presentation, and that we have now opened the floor to questions. If you don't want to unmute yourself, you can put a text on the text box and ask a question.
Yeah.
Okay, Prof, maybe I will start.
Yes, go please.
I really enjoyed your presentation, and one of the questions that just sprang into my mind in the beginning of your presentation, and also just towards the end, you spoke about how expensive internet, the cost of internet is in Africa, right? And then you later on explained all the different questions we can ask using genomics and AI. I just wanna. Naza, can you mute? Okay, thank you. Yeah, so I just wanna ask, if you look at Africa and the state we are in currently with our academic institution, how many years do you think it may take to actually reach the level where we are able to compete at an international platform when it comes to genomics and asking the same questions that the rest of the world is asking?
Yeah. Thank you, Lile, for the question. Look, I don't think it's that complicated. Um, let's give an example just to make the picture clear. The sequencing of the human genome that was done in 2001, was completed in 2001, took 13 years with the collaboration of all the countries of the world, at least all the developed countries of the world, including Europe, United States, Japan, China, and all. The same labor could be done actually in one day by one scientist in 3 or 4 hours. So why this? Because the jumping of the technology. So we are living in an era where the technology is advancing in really an unimaginable way, that I think if there is a will, anything could be done in short time. I mean, there is no limit to the technology. I think African countries, they have the money, they have the biggest diversity in the world. They have all what they need. If there is a political will to sequence thousands of genomes, including humans and non-human species, I think it could be done in a short interval of time. And I believe there is no complexity to that. It's very doable, I think. Thank you.
Thank you, Prof. Just a follow-up question on your answer. You say that if there's a political will, and I think that is actually the crux of the matter, that maybe we don't have a political will. And I don't know if Professor Amadi is still here to actually tell us if there is a political will when it comes to making this happen in our continent. I don't know. I don't think he's here anymore. Yeah. I think he's signed out. Okay. Yeah. So after my question, Prof, if you are able to answer it, I will hand over to Stephanie and then to Lawrence, as well as to Zama. They can go in that sequence and also ask their questions. Anyone who doesn't want to go on live can actually just type their question and put them on the text box and we will read them.
Yeah.
Please go ahead, Stephanie. Please go ahead and ask your question. I see you've raised your hand. You can ask the question.
Can you hear me? Can you hear me? Good afternoon.
Yes, I can hear you.
Yes, we hear you.
Thank you so much for this impactful
Thank you.
presentation. My question is, you made mention of maybe subsequent researchers, instead of going ahead to test for a particular gene, can actually get data from the data bank. My question is, is this still necessary for someone who still wants to go ahead and test for the gene, to test it and maybe compare it with what we already have in the data bank?
Stephanie, could you say it again, please? Because the voice, I'm not hearing very well.
Okay, sir. Can you hear me now?
Yes, go ahead, please.
Okay, you said during your presentation, you said that it is possible for someone to actually get data from a data bank when you are talking about Alzheimer's disease.
Yes.
So my question, for example, a researcher that wants to go ahead and test for that gene, him or herself, is it possible, or will it still be relevant for the person to test for that gene and peradventure compare it to the one we already have in the data bank, or will it be a waste of time?
Yeah. It's if I understand the question, Stephanie, you are asking if I have an expression of gene myself of my experiment, and I would like to compare it to the other database to see how my gene expression is changing in comparison to the database. That's the question, Stephanie?
Yes, sir. Yes, sir.
Yeah, of course. It's not, it's I think the straightforward application of the database. If I have a gene expression of my own experiment, the first thing I want to understand is how it is changing in comparison with other people, other patients, or other healthy people. How it is changed? Is it the same? Is there any characteristic intrinsic to the population I'm studying, or it's like the other? So I think this is the most straightforward application of having the genomic information in the database. I think Lawrence is raising his hand.
Yes, Lawrence, please go ahead.
Okay, thank you, Alejandro, Lulu. I'm grateful. I want to first of all commend the presenter, the prof, for that wonderful presentation on genomics. I just have few observations and questions that I want to raise. Actually, I'm from Nigeria, and then I'm actually working on Alzheimer's disease and trying to go into the neurogenomics. However, there are challenges here in Nigeria in terms of we have few laboratories that we can collaborate to work on neurogenomics. You mentioned a few things, a lot of things, not even few, in terms of functional neurogenomics, metabogenomics, cognitive genomics that we can do. But the problem is that I don't know, maybe you can have, with time, organize a workshop that have to be on hands-on training, you know, and of course, to help us so that we can have a lot of data bank for Africa, because African, we have issue with data bank for most of these things that you discussed. And I think nobody will do the work for us now. But for us to even create, to have more pull to data bank, we need to also have a lot of hands-on experience so that with time, we can begin to do some of those things on our soil. So I don't know, maybe in the near future or sooner, you're going to organize a work and hands-on workshop training.
Yeah. Okay. That's the question, Lawrence?
Learn on the, hello, can you hear me? Hello?
Now, yes.
Hello?
Now, yes. But before it was just...
my question is very clear, Lulu. Thank you.
Okay. Thank you, Lawrence. So, I'm aware that there are few laboratories, either in Nigeria or all the African soil, that do this. So for that, we would like to have more platforms for that. For the workshop, what we can do, because as I mentioned in the presentation, I'm the North African coordinator of Africa BP project. And within this project, we organize some workshops. So I can ask the coordinators if scientists from other communities, for example, here from neuroscience, could join those workshops and could learn how to access database now.
All right. Thank you, Prof. If you can send the link of the workshop, will be interesting. And then, of course, we will.
I send you the link. I send you the link to the project, the website of the project, and there you will find all the information, even the email, the coordinators, and all. I will speak with them, but you can do it to yourself to emphasize and to insist, and hope that something will come out from this.
Okay, please send to the coordinator. The coordinator will get across to us. Thank you so much. Thank you. All right.
You are welcome, Lawrence.
Thank you.
Zama, are you still there? Do you still have a question for Prof? Zama, you have your hand up, but I think you didn't connect with Wi-Fi. There's no audio on your hand. Can you text the question? Can you put your question, Zama, on your text box? Okay, now you have a voice. Do you still want to ask the question?
It's in the text box, so but I can ask. Prof, you talked about how we can associate phenotype with especially the SNPs. Some of them have been identified at data bank. However, as you have rightly mentioned, a lot of these studies have been done abroad, different populations, and their SNPs may not necessarily apply to our population. And sometimes you may think that we don't have the SNPs, but we actually do. We just don't have the funding to try and identify maybe sophisticated tools like genome-wide association studies. And so would it be a wise idea to actually collaborate with international researchers who may maybe approach us as African researchers, as we have access to these populations that haven't really been genotyped, that haven't been looked at? Is it worthwhile to try and do it ourselves? considering the cost. I mean, it's in the millions trying to even get a good enough sample size for you to be able to do analysis on those samples. So what would be a way forward for an African researcher who actually wants to get into genomics?
Thank you very much, Zama, for a very interesting question. So, of course, acceding to the database, it's good, but it will not resolve the problem for the African population. For that, we need to have our own database, because as you mentioned, there is many SNPs that are responsible for some diseases, for some pharmacogenomic characteristics, but they are not the case in the African population, because it's a different population. So, and all the precision medicine and the works that was done and that now exist in the databases is based on Caucasian white populations mainly. So, of course, this is a big problem, but as I mentioned it, if there is a consortium, a big consortium, and a big will to sequence the African genomes and to make databases that could be used later for precision medicine or for getting the good SNPs for each population, I think it's possible. I mentioned in the first part of the presentation the case of Africa BP, which is started from nothing and now it's going very well. Many genomes have been sequenced and more it's coming. So just by contacting the founders through all the world and getting the information. This is important because we hope that the African Union will pay the most of the money just because of the to have the ownership on the data and to have the ownership within Africa. And of course, you can ask for additional funding from the outside. And the outside, they can give money, not just because they give money, as we say, for the beauty of our eyes, but they give money because they know that there is a huge diversity in Africa, and they will use some of this diversity in the precision medicine chips that are already designed in the Occidental world. So they need even this particular mutation or information that they can get from the African population. So to make it short, yes, there is a huge investment to get the correct and accurate information concerning the African population by sequencing genomes of a maximum number of African people. But it could be done. It could be done by making a consortium, asking for money, and if, for example, companies, they will not pay for the sequencing, they will pay later for the introduction of precision medicine in the African countries, because introducing precision medicine in African countries is very attractive to the international companies. So they are ready to help in identifying the good SNPs and the important SNPs, because later they will win by introducing all the infrastructure of precision medicine in the African countries. So there is many scenarios, but I think it's possible, and we need just to go for it. That's at least my opinion.
Thank you, Prof.
You're welcome.
Thank you, Prof. Thank you, Zama, for that question. And actually, I didn't realize that people put their answers on the Q&A section because I saw that Tandega said we must put it on the main text box that everybody can see. So there's a couple of questions here for you, Prof, as well as comments. I'm just going to read them out. Stephanie says, thank you. It was very impactful presentation. And then an anonymous person says, thank you for the great presentation. Can I ask if African BP is involved in genomics studies training for upcoming scientists? How can one join for the Africa BP?
Yeah. So yes, we have Africa BP project, and we have other, let's say, institutions with it, like African Open Institute. We are trying to build uh, African Institute of Bioinformatics. So yes, the training and building capacity is a big milestone of Africa BP project. And you can be a member of Africa BP. It's not that I will put, I don't know where is the chat box because I can't find it. I don't know
Yeah.
if I'm
So, Prof, you can send it on our via email, and then
okay.
we will post it on the
That's
SONA
great.
website.
I will send it to you, Lile,
Yes,
via email, so you
yes.
can share it with the participants. So yes, you can be part of Africa BP. Just please go to the website, have a short read, and you will understand all the procedure.
Yeah, thank you, Prof. Another question is also, how comprehensive is the phenotypic studies in Africa based on few data?
Yes, of course, we cannot have a deep understanding of change of phenotypes, either disease or other phenotypes, on the basis of few data. That's not to be the solution. What we can do, let's first say that there are some countries that are doing many important things, either in South Africa, in Kenya, in Morocco, in many other countries. But it's disparate data. So if we can put this together, this is already, it will be something in which African scientists could start to rely. But to answer your question, yes, we need more data. We need that data. We need consortium about the sequencing of the African genomes. We need that. That's urgent thing that African leaders could tackle to solve the problems. But waiting for that, there are many works that are done in many countries through the continents. If you make some effort to get this information and to contact the people, maybe you will have something on which you can rely, at least in this period.
Thank you, Prof, for that answer. I'm just going to read the last two questions so that you can answer them at the same time, because we've run out of time now. So another anonymous attendee says, Professor, if you can elaborate on which pathway DNA, RNA metabolism, protein to choose on how to research a topic such as the cause of a neurodegenerative disease, do we make hypothesis when it comes to DNA expression first, or do we look at protein aggregation, or what exactly?
Okay.
And then
Good.
the last one, did you get this one properly?
Yeah, I think yes. If not, correct me, please, Delay. Yeah.
Okay. And then the last one is, are there any advantages on focusing on the mitochondria's DNA when first looking at a neurodegenerative disease?
Okay. So for the first questions, I mean, which approach of genomics to use either structural genomics using DNA or microRNA or proteins? As I mentioned, it depends on the scientific question, because the insights we can get from DNA are different from those we can get from gene expression, like from mRNA, are different from that we get from protein. So let's take an example. If uh, if for example I have a disease, let's take for example the onset of Alzheimer disease, the onset of Alzheimer disease, and let's say how the genome changed before and after the onset. What will change actually? It's the gene expression between before and after. So this is, or let's say, if we ask it a question, how I can discover the genetic basis of the development of the Alzheimer disease. Let's start that there is a person for which the Alzheimer disease started since 2 months ago. So, and I can take mRNA, study the gene expression in this person, and after 1 year, I can study the same gene in this person, the same person. So I can see how the gene expression changes between these two periods, between the early disease, the early Alzheimer, and the late Alzheimer. So in this case, I will not go to look for DNA because I will not change. The DNA is fixed along the life of the person. So I will change, I will look for the expression of the gene in the same. So I'm not interested to look to the DNA. Now you can ask me, should I look to the mRNA or to the proteins? It depends, because in some scenario there is a big correlation between the expression of mRNA and the protein. It could reach 99%, but in other cases it can reach only 20%. So if you look to gene expression, you could not have an idea about the protein expression. In this case, you may perhaps do either proteins or both to have a clear vision of what is happening. So just to summarize it, it depends on scientific question and on the biology of the disease you are doing. So that's the main my main answer. And the second question is about mitochondria, if it's important. So yes, of course it's important, because mitochondria, it contains the breathing genes and respiration genes, and this is, I think, it could have a big implication in many diseases. And second, as it has maternal heredity, not paternal heredity, it's good to understand something that is maternal when using mitochondria. I think this is the biggest use of mitochondria, of looking to mitochondria in the context of biology or neuroscience in this case.
Thank you very much, Prof, for an amazing talk and a very enlightening question and answer session. I think we probably need to invite you again, because I see that there was a lot of receptiveness to from the audience. Even the attendance was amazing. So I think we probably need to have a second session on this talk. Thank you. I can see there's a lot of hands clapping. And to the audience, anyone who, I saw someone asked for the link, all the links that Prof has spoken about, including the link for training, the link for Africa BP, and the recording of this particular webinar, it will all be posted on the SONA website. So please visit the SONA website. Tandega will post everything. It should be live before the end of tomorrow. Thank you very much. All the best, Prof, with this amazing research, and all the best, everyone. Thank you for attending. Goodbye.
Thank you very much, Lee. Thank you, all of you.
Thank you. Bye.
Bye.
Thank you. Bye.